be talking about the word inoculation. What does it mean to inoculate? When you say you want to inoculate in the lab, what does it mean? What does it mean when you say you want to inoculate? If you are registered, if you are doing your lab now, I believe you should have a response. What does the word inoculate mean?
Like to sterilize.
Okay. Okay, I see something on the chat as well. Put the bacteria. Okay. Now, do you notice what do you know what you do when you use the word inoculate in the lab? Okay, let me ask you this. Look at this. Do you remember this? Do you remember? Have you ever used this in the lab? Okay, good.
Yeah, we use.
Good. Do you remember what you do with it?
Yeah, we use that inoculating loop to transfer that what we call this to the culture to
Exactly.
crop the microbes.
Thank you. I'm seeing the chart also. Thank you. That's the meaning of inoculation. So it's simply to transfer bacteria from one medium to another. That's what you do when you inoculate. You remember that when you inoculate, before you start it, most likely you flame your loop. Inoculating, you flame it. Flaming it is the purpose of the flaming is to ensure that you are sterilizing it so that it's not picking up any other bacteria or mixing it up with the one that you have and not contaminate your media. That's the purpose of inoculating. All right, so culture, what is culture? When you say you want to culture your bacteria, actually culturing your bacteria is simply like growing your bacteria. And we use the word cultivating, like you want to cultivate it, you want to grow it. When you say that you want to cultivate something, you want to grow an habit, you want to grow a quality. So that's the way culture is. So it's the heart of cultivating microorganisms or the microorganisms that are cultivated. So we call it culture. So you can say, oh, my bacteria culture, my culture. All right. You inoculate something now, you do it, and then you leave it there for a week and come back in the next class. That's your culture. All right. Now, then talk about the culturing medium, meaning that the medium where you put the bacteria in. So you can use a algae medium, agar. Right now, inoculum is of microorganism introduced into the medium containing nutrients. Now, inocula obtained from various sources. You could get them from environmental specimen, you could get them from clinical specimen, you could get them from stored specimen, depending on it. You get to the lab, your professor gives you a test tube, and he say this one contains E. coli. You have another one, he say this one contains diphtheria. You have another one, you say this one contains pneumonia, the bacteria that cause pneumonia. So it's different. It could be from stored specimen, could be from clinical specimen, meaning that from patient that are, for analysis, right? And it could be from, you want to test water, environment. That will be environmental specimen. All right, let's go. So those are talking about the type of locations of specimen and where they are with the way that they are collected, right? So take note of that. Now, obtaining pure culture. How do you obtain pure culture? Of course, you will have done this in the lab by now, where you were trying to purify your culture to isolate your culture, isolate a bacteria. Now, our cultures are usually mixed up. You could have mixed culture. Mixed culture means it has multiple types of bacteria in that same culture. So how do you isolate it? So progenitor is a term used for colony forming unit, CFU. Aseptic technique prevents contamination of sterile substances or objects. Now, what do you do when you want to isolate culture? We use two medium. We use a strict plate, which is what you do. If you have done it in the lab now, you take your inoculating loop and then you sterilize it, let it be cool down a little bit so that the heat does not kill the bacteria when you put it. So you let it cool down a little bit, then put it inside the bacteria. then you get a loop out of it. When you get the loop, then you're going to put the loop in the dish that you have, then you streak it like this. You do this as well. That is your, that is your streak method. This is your streak. We say you are streaking your plates. Right? So your bacteria, it could be a solution inside a test tube. You can have your E. coli inside a test tube, you get it out. Or you could just pick a colony from a plate as well and then streak the plate. That is streak method. Pour plate is what you do when you see, you have this bacteria in the test tube and then you pour it into the plate. Don't mind me the way I drew the first one. I know that's not the best way to draw plates. I just want to show you. Neither is this one the best one too, but at least you can see. So the pour plate, you pour it into the plate as well, depending on what, I mean, I'll still show you some later, but that's, all right. So you see here now, you've got a colony, you just got a colony. This is your inoculating loop. I'm doing this for you are not in the lab this year, semester, this inoculating loop. So you use the inoculating loop after flaming it, get a colony of bacteria, transfer it here, you see, you streak. Sometimes we divide the plate, we divide the plate into 4 in a way that you have it in different areas. So what do? Look at it. You see, this is the first one. So you streak the first one this way. Now, so the first one, you streak it like this, you streak it. Then you just go from this first one, just take, in quote, 1 from here and go to the next area and streak it. Now, what did you do? You go from here again to the next area and streak it. You go from here again and streak it. What have you done? You will notice that at the end of the day, look at it now. At the end of the day, you have more streak in this area. I mean, you streak this area the first time, right? Then you came here, then you came here, you came here. Now, look at what has happened here. Now, you notice that there were a lot of concentration here. Let me see if I can change this color. You will notice that on this area, you seem to have more concentration there, even though, you see, now here is still close, not as much. I mean, it's still close to what you have here. But what do you notice here? In this area, you have little now. In this area, you have quite less. What has happened? You are gradually isolating this bacteria. So let's say there were mixed culture here. There were mixed culture here. There were mixed culture in this place or place before. You took just one out and then you streak it here. You then just went ahead and continued that streak. Now take note, this streak is not like this. You think it's not like this? No. It is that you took one bit out of here and then streak it on this area. Then you took one bit out of here and streak it in this area. Then you took one out of here and streak it in this area. So in actual sense, you have reduced the population or the concentration of bacteria from 1 to 4. And that's what you notice also in the plate after incubation. Right? So this is one of the way we isolate bacteria, such that you now pick a colony from here now and then put it on a clean plate. Then you notice that you have this particular bacteria here. So that's isolation. Okay, this is pour plate. So you see, that's pour plate. You do 1 ml from here. Again, this also we call it serial dilution. We call the process like serial dilution. In essence, we are also trying to isolate the medium. So what do you notice here? You took 1 ml from here. This is the sample where you have a lot of them. You took 1 ml from here. Now you put 1 ml in this 9 ml broth, meaning that this is a medium. It's a broth, it's a medium. Now, then the next thing is this. You put that 1 ml here. The 1 ml mixed up with 9 ml broth. So we call that 10% dilution. Then you put it in 10 ml broth, took 1 ml from the 10 ml broth into another 9 ml broth that was here. Now, this is already 100-degree dilution. Now, you took from here again into another one that is 1000-degree dilution. So now, look at it. When you plate this one now, and you incubate it, what do you notice? You have different bacteria. You can see different type of bacteria there, and quite a lot. But when you did this, what do you notice? You saw fewer colonies of those bacteria. So what can happen now, if we actually want to get extremely pure culture, because here you still have these, here you, oops, wait. Okay, good. So here, you still have this. You have another one, you have the blue. So now it's easy to isolate this bacteria now to say, okay, I want a dish where I'm going to only have this one. Then you can transfer this into a dish and see if it's going to grow. You can transfer this into a dish. And then one of the things you can now do, you can now use a kind of medium that only selects the bacteria you want. So when you put the bacteria, when you put all of this in a medium, it's only the one that you need that will grow, right? So again, this is pour plate. This one we call it the pour plate because you are just pouring it. And then the other one we call it the streak method. Are you with me?
Yes.
Right. Thank you. Oops. Okay. So then obtaining pure culture, which is what I've been trying to talk about ahead as well. Now, other method that you use, some fungi isolated with streak and pour plate method. While some protozoas and multi-unicellular algae, you can use dilution of broth, like we said. You can identify pick a single cell, just like what I showed you the other time, and then use it to establish a culture all by yourself alone to grow that one such that you are sure of the pure culture that you already have. Now, you can also use culture media, like I said. So majority of bacteria have not been grown in culture. Majority of prokaryotics have not been grown in culture medium. Variety of liquid and solid media, they are used to culture microbes. So we have the nutrient broth, which is what we use most of the time, liquid. Then we also have the algae, which is one that is most common, solid media that we use majorly. Now you put a petri dish and also a slant tube, which is what we use normally. All right, so that is it. Now, when you say slant, you will always see this in the lab. You see this is slant because it's slant. All right, so this is a medium, of course, but we call this slant. When you see it like this, it's different from the one that is just like this or like meniscus. It's different, right? Again, they are just different medium or different media. Now, the culture media that we have 6 types. We have the defined media. I'll be talking about them. We have the complex media. We have selective. We have differential. We have anaerobic, and we have transport. So I'll be talking about the different media as we go. Now, the ingredient that I experience that you're going to see in medium or in the media, particularly for E. coli, for instance, is going to have this nutrient with this proportion. All right, so it's going to have glucose, it's going to have sodium hydrogen sulfate, phosphate, it's going to have potassium phosphate, it's going to have ammonium sulfate, it's going to have magnesium sulfate, it's going to have it's hydrated, then you're going to have calcium chloride, iron sulfate as well. Now, those are the composition that you see in that will grow E. coli appropriately. Now, culture media, we'll be talking about each of them that we listed earlier Now, complex media. Now, when you say complex media, it's simply, another way you could think about it is mixture. Now, what it means is that you can't say this is the name of the media. You can't say this is the name of the nutrient that is there, because it's made up of a couple of nutrients. So you don't call it one. It's made up of multiple nutrients that makes it up. So nutrient commonly derived from breakdown of yeast could be there as breakdown. Beef will be part of the components. Soy and proteins are there. Now, they support growth of wide variety of microorganisms. Why? Because they are mixed themselves. It's a lot of nutrients that could serve different organisms. So a lot are there. So it's just like if you put different organisms, like a mixed culture there as well, there is resources, or there are resources for different organisms themselves. All right. Now, so they are useful when nutritional needs of an organism are unknown. So if you don't know what bacteria you have, you just have this bacteria you don't know, you can use a mixed culture for them. You can use a complex media for them that consists of different nutrients that can feed whoever or whatsoever. All right. Points where you use a very definite medium, which we call simple medium. It's one of the simple medium that have just a specific, is you have this mixed culture, but you don't need all the other organisms, bacteria. You only need one particular bacteria. Then you put that mixed culture inside a media that you know only favor the bacteria that you need. Let's say you need E. coli, for instance. Now you're going to put it in a bacteria in a medium that only supports E. coli. So what's going to happen is, over time, the ones that is not supported will die, whereas it's going to be E. coli that will keep increasing. It will keep growing. So as you transfer it, you end up now having a medium that is only E. coli, like a plate that is only E. coli that is in there. All right. Now, the second one that we have is a selective medium. The selective, all right. So you look at it, they contain substances that favor or inhibit growth of particular microorganism because they are selective. They don't sponsor the growth of all the bacteria. They have something that can grow some. They have something that can inhibit particular bacteria you want to. Now, this here now, for instance, now it model what you see in selective bacteria. So you see selective medium rather. This is specific for pH 7.3. So if you put a bacteria or any organism there that cannot survive that pH, they are not going to grow. They are going to start dying because they don't have support. And then this is pH of 5.6. All right. So fungal colony could grow here, but you will not see bacteria thriving at 5.6. You will not see them. Now, enrichment. So we have these media, we call them selective as well. And then among the selective is the enrichment meat culture. Now, the enrichment, what it does is that it's going to favor the growth of a particular bacteria. It favors. So you see, it's selective medium used to increase small number of a microbe to observable level. So it will favor the growth of those that it want to select. So we call it enrichment. Then we have the cold enrichment, which are just for the cycrophals, the bacteria that are cycrotolerant. All right. So this blood agar. All right. Now, differential medium, this very interesting medium. Now, what they do, so it's different bacteria growing on a medium, on a media distinguished by what? Number one, presence of visible change in the medium. So you will notice that there are changes in the medium, and then difference in the appearance of the colony. colorations, all of that tells you that this is different. So the color, the concept of the media is to differentiate it for you anyway. So you see, in this place now, if you use this, now, if you use, there is a way that we use this to check organisms that use carbohydrate. So we call it carbohydrate utilization soup, right? Now, so organisms that use carbohydrate, the color will be different based on their metabolism. It's just like if you do this experiment when you are looking at, I'm trying to remember this now, but there's this experiment if you are using, if you use organisms that use carbon dioxide, for instance, will turn purple. The one that doesn't use will be yellow, thereabout. So there is a way that you can use media also to test. If you put the organism inside the media for about 24 hours, at the end of the day, their color changes will point to, okay, these are these organisms that are here. All right. So in this case now, this is here, no fermentation, so the color remains the same. Now, acid fermentation happen with gas. All right. So those point what kind of bacteria that are there. And you already know the bacteria that does acid fermentation anyway. So that will be the way to check different bacteria as the case may be. So it's differential media. All right. So these are composition of, we call this McConkey medium. And those are the composition of the components there. Then we have the blood agar medium, which we use very well. Okay. Now these are differential medium. Now, so if you look at this now, you have E. coli on this side, you have Staphylococcus aureus on this side. This is a nutrient agar. So you're going to check what's going to happen. Is there growth after about 24 hours? Yes, there's growth. You can see the green. And of course, the color here, this nutrient agar does not differentiate it for you. But then you have McConkey agar here, which is not going to allow the growth of Staphylococcus aureus. So that's what I was saying. These are selective media, for instance. Now, it's going to select which one and then differentiate them for you. Like, so that this organism is present, that's where there's growth. So if you don't have E. coli here now, that means nothing will grow. All right. That's the way they work. Now then, if you the same thing, McConkey agar now, because this is I think it should be because it's acidic, it's going to turn yellow now. So it turns yellow, this one remains red inside the medium as well, right? Now, whereas this is Salmonella enterica. So those are ways to test, to take note of the bacteria that are present. We can use to check the medium where they are going to grow or where they cannot grow. And then we can use medium that will differentiate them like this. This is selective media, for instance. This is differential media. So this one will be selective. This will be differential, selective. So those are the ways we use media or medium. It's 337. All right, let's go. Let me see. Transport medium. The purpose of transport medium is simply to move bacteria and then to prevent contamination as you transport them. That's the major thing for transport medium. Animal and cell culture. Now, these are mediums that you use to culture those animal cells. And the reason we talk about them is that, of course, those cells are meant to grow naturally, right? So when you have those culture, we call them, it's more like an artificial medium where we can put them. And of course, you know that viruses don't grow by themselves. They cannot survive by themselves. They don't do metabolism by themselves, which is the reason the conflicts in identifying them as living thing, right? because even though they do all of those characteristics of living things in quotes, but they don't do it by themselves. They do it in the host. If you pull them out, they're not going to do any of those functions. So low oxygen culture, that's carbon dioxide incubator. They mimic the environment of certain body tissues, right? Now, candle jars, we create environment with low oxygen and high CO2 level, right? Now, preserving culture, of course, that's refrigerator. All right, we have the deep freezing, which can store bacteria for years. And then we have the lyophilization, which are able to store for decades. Please excuse me a little bit. So, all right, so the concept of course, the concept of lyophilization is like deep frying. So it's more like a free drying rather. It's a way of free drying something. So it's like free drying. Freeze drying. Don't mind me. Is this? I'm still getting used to this thing. Freeze, like freeze, drying. So it's a way of freeze drying, right? So the concept is that it drains water from the bacteria, and then it's going to make it frozen. So it drains water from it, and of course that makes it last for long. All right. Now, when I say that bacteria grow, the way they grow, I use the word splitting, that it's more like they are splitting. We call it binary fission. Of course, you know that fission is different from fusion. Fission is to split. Fusion is fuse together. So this is what we use. We use fusion. For bacteria, they do fission, which is to split. They split into two. So one divide in half to produce two daughter cells. So that is fission. All right, let's go. Right? You can look at that video there. You won't be able to see it now you won't be able to hear it. So this is what it looks like. It's just like you're thinking of mitosis anyway, but it's called binary fission in bacteria. It's more like that. So the simple way to think about it is like binary fission. All right. It's okay. So so you see, this is what we are talking about. It's like fission here, splitting. This is a bacteria, then it's as produce, duplicate its nucleus now, then goes through the process, eventually produce two cells. So that is just like you think about mitosis. All right. So then time required for a bacterial cell to grow and divide, we call it generation time. All right. And it's dependent on chemical and physical condition in the environment. If there's resources in the environment, they do that. If not, they can go into endospore formation or hibernate. All right. I don't think I need that for. Okay. Now, when you think about growth, we have the point that we call the lag phase. We have the log phase. We have the stationary phase and phase. Now, the lag phase is more like acclimatization period. So you think of acclimatize. That is the lagging phase. It's more the time they are adjusting to an environment. That is the lagging phase. So we call it the acclimatizing or the adjusting phase when the bacteria is still adjusting. That's a lagging phase. Now, the log phase is when the bacteria is growing. That's why it's called exponential phase. So it's dividing very well. Division is happening there very well. So it's the time when the bacteria is actively dividing. That is the log phase. So lag phase, there's no increase in growth, no growth. Let me see if I can make this to be better. Okay, it's not letting me clear it. So in the lag phase, like I said, no increased growth, no increase in the number of cells. There's no increase at this level. I can't hear anything. Really? Can others hear me? Just to check to be sure of what is happening.
Yes, we can hear you.
Okay.
Yes, we can hear you.
Okay, please, you want to check your volume, or your device, maybe it's on your hand there. Okay. Please confirm if you can. Once you can hear me, just let me know on the chat. So the lag phase, like I said, there's no growth. The bacteria are just there. They are trying to adapt to the environment. It's just like an adjustment phase. Okay, great. So it's just like an adjustment phase. But the log phase is where you have an exponential growth. There will be exponential growth, which means there will be increase. Because I told you already, growth simply means increase in number. So here, there will be exponential increase in the number of cells. Growth in bacteria means increase in number, not increase in size. All right. So the number of cells will increase. Now, stationary. In the stationary phase, majorly what happens is that the rate of, so I'm going to say death is equal to division, more like that. So the rate of death is equal to the rate of division. When I say division, I mean cell division. So that means it is stationary. There's no increase, no decrease. The number of cells that are dying is equal to the number of cells that are alive. And of course, the decline stage is when there's no more division, there's just death. So we say that there will be decrease. There will be, just the way that there's exponential increase here, there will be exponential decrease here. Because at this point, no cell is dividing, all the cells are just growing, are just dying. All right, that is that. Please take note of those phases and their peculiarities. That's straightforward. Lag phase, there is no growth. Acclimatization is what is happening there. Log phase, there's exponential growth, which means increase in number, not size. Stationary, equal number of growth, equal number of death. Death, exponential decrease. All right, chemostat is used to, of course, you can, even if you don't know chemostat before, you know thermostat. And you know what thermostat does is it shows the statistics of temperature, it regulates temperature. So what chemostat also do is to regulate the chemicals. All right, so chemostat, think about it that way. So it's used to maintain a microbial population in a particular phase of growth. Now we have the open system, which we always acquire that it will always require that you had fresh media into the system. So it's more like it's open, so you can recycle. Think about it like open in the concept that you can breathe in and out. So we are open system. So you are breathing in and out. So the same way, you can come in, remove old media, put the new media there. So we suction it. We use it like a suction. And then we take out the old media and then put in new media on the, so we call that the open system. All right, so so this just showing you like what a chemostat looks like. That's the culturing vessel. That's the overflow tube where they can flow out as they overflow. That's the culture itself there. That's the sterile air or gas, other gases that can come in. And then you have the fresh medium inside there. Okay. Now, how do you measure this? Now, when you want to measure microbial growth, you are still going to likely do it in your lab, but we use cytometer to do that. Now, because what we are doing is simply to count the cell in culture, right? So we graph it. Now, so estimating the number of microorganisms is useful. Now, it determines severity of certain infections. Now, it determines effectiveness of food preservation technique, and it also helps to measure contamination of water. It also helps to evaluate disinfectants and antibiotics as well. It's something we do in the lab, in microbiology lab. I mean, we do it particularly for non-science major. We use antibiotics, and then we put the antibiotics. Sometimes we use these this little disc, antibiotic disc, and then we put it there and see bacteria will go around it. We measure it, all of that. So those are ways to count bacteria as well. All right, this here is the cell counter. You are going to likely do this also in your lab, depending on your results, right? So we use this, then you look at this. This is what you have here now is what you are having here. So you take this under the microscope, you will be able to see this. This is like slide. So you take it under the microscope and observe it, then you can count the number of cells looking at the cubes, each square, and then count the number of cells like that. That's where we can count cells. So in a way, it's more like this now. So you see, you have the whole thing. This is here. Then this one now is what is magnified that became this. So that's what is there. So how do you count this now? So if you count this now, then you can use it to estimate what you have in total. So we can do direct method. So measuring microbial reproduction, we can use direct method without using incubator. We can use, we call it culture counter, which is what I just showed you now. We count it and then estimate based on what we have counted in each cubes. Then you have a flow cytometer. What flow cytometer does majorly is that it detects changes in light transmission in the specimen. Let me see if I will still show you something about that more. Direct method that include incubation, that include that you have to incubate the bacteria. We do serial dilution, which I explained earlier. Then we count it based on the plate count. We count the cells that are variable, the cells that are alive, using a plate count. We use membrane filtration. Again, like you see, the most probable number. We can only estimate based on, because you're not going to count everything. You just count and then use a formula to calculate the proportion that you have. All right. Again, this serial dilution, I already showed you that earlier. All right. So serial dilution, okay, this is what I was telling you, that this like 10%. So that's 1:10. It means you have one, you got one from your specimen, you put it in 9 drops, 9 ml drop broth rather. So 9 ml broth, and this is 1:9:10, meaning that in 10 ml, you have 1 ml bacteria in 10 ml. That's 1:10. Then you do the same thing. You took it here. So this is that 1:10 is here. Now you took 1 ml from the 1:10 that has been diluted 10 times. You took it here and then put 9 ml of broth again. That is 1:100 already. You do the same thing from here. That's 1:1000. You do the same thing from 1:10,000, 1:1 million. So, 100,000. So by the time you transfer them into the plate, you incubate it. You put 0.1 ml of each of them into the plate. You incubate it, see what has happened. Here, you have a lot, too numerous to count. So we just call it TNTC. Now, this is also too numerous to count. Now, here you have 65 colonies. Here you have 6 colonies. Here you have 0 colony. All right. So that means if you want to do an experiment now, this is what can give you something. Here you will get nothing, just like you can see there. All right. So that's serial dilution, which is what I talked about the other time. That's one of the ways that you isolate bacteria. All right. This is filtration. It's more like you're using a filter. Think about using a filter paper and getting some out of it. That's concept of filtration. All right. Then you put it in colony and see, put it in plates and see what you get from it. Right? This is still the concept of, I don't think I need to explain it again. Let me see. Sorry. Okay, so the indirect method, we use turbidity. Now, let me see these here. Sorry. Okay. I'm trying to see if any of them, these also explain turbidity in a way. But now, what turbidity does majorly is that it's an indirect method. Like I said, it's not a direct method to count. Like we use flow cytometer, cell counting chamber, those are to count. And you will do all of this in the lab, which will make it better clear to you as well. Now, but what turbidity does majorly is that it measures how cloudy the solution is. So if the solution is cloudy, the cloudy the solution is, the less cloudy or the more cloudy means there's a lot of bacteria there. If it is, okay, so you see often the more to be turbid a culture is, the greater the concentration. So in a way, sometimes in the lab when we pick a test tube, we can tell if it is so crowded. If it's a lot of bacteria that are there and that you still need to do isolation quickly or to do serial dilution so that you can know instead of wasting your time. So just by looking at it, you don't need to count most of the time. Just by looking at the test tube, you can tell that there's a lot of population of bacteria here. So that's the indirect method. That's what the turbidity does. So turbidity and the use of spectral photometer. So this is you see this is clear. This is turbid a little bit, right, compared to this. So that's what the turbidity can do. So you have source of light to look at that, and then you can assess it to see if it is turbid or not. Then you can use the spectral photometry as a way to check as well. Now, the indirect method is still metabolic activity is another thing as well. If their metabolism is high, that's something. Dry wave molecular method that isolate DNA sequence from unculturable prokaryotes. And this is an example of what I was saying. You can just take them and put them in a medium, culture them, and you discover that the DNA, the one that cannot be cultured, will be separated on that note, right? So that's that. Now, let me see.